This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 77, recorded on September 18th, 2014. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
Sporting the Steve Jobs look.
Oh, how nice of you to say that.
Why? Because he's dead?
No, not because he's dead. Is that what the Steve Jobs look is? You have to be dead to look like Steve Jobs? No, I think
jeans and a black turtleneck.
Well, this is. I've worn this many times
Professors
before.
do not wear jeans and black turtlenecks.
You never said that to me before, and I've worn this outfit several times here.
I always think of some way to insult you. Why not, right?
Well, it's hard to do, you know.
Dickson, we're already halfway through September.
We are.
This is pathetic.
I know.
And you know what the weather is today?
I do.
You know, it's
delightful out.
It's delightful. 23C,
it's
which
marvelous
is like in the
there.
low 70s.
Yeah.
And the humidity is way down in the 30s.
That's exactly why it's delightful.
No wind, partly
it's
cloudy.
perfect.
So you're looking out the window.
I am.
Behind me. What is it? Are there clouds in the skies?
Yeah, they're scattered clouds, as they would call them.
And it's
a
chilly
Georgia
now.
O'Keeffe sky.
In the morning when we leave home, it's chilly, right?
A little bit. And you can see little brown edges around the leaves now. So
they've already fallen in my town.
Fall is coming in. We haven't had rain in a while, and that's another reason why they're falling off.
Yeah, and some years they fall really
That's
late.
Right. You know,
and
the
some
poet,
trees are more susceptible to early falling than others.
This is all true. Do you know the poet William Carlos Williams?
I do.
Really?
Very much. He lived in Passaic, New Jersey. He was a physician. My father knew his son.
Really? Well, he taught at the school that I got my degree at. Fairly
Useless?
That's not the name of that.
Fairly Ridiculous.
You know, you're really pushing the limits here, Dr. Racaniello.
I know a lot of
just because
listeners
you went to Cornell
probably went to Fairleigh Dickinson. I don't mean to insult them.
Now, we used to call it Harvard on the Hackensack. But of course,
tongue-in-cheek, right?
Tongue-in-cheek, yeah. We realized it was a great school, though, for many reasons. And one was that it was convenient, and two, that it was affordable. And three, believe it or not, I got a very decent education there.
Well, look, you're
According
a Columbia
to me.
professor. You did fine.
That doesn't mean anything. I had to pull myself up by my bootstraps.
Doesn't mean anything that you're a Columbia professor.
No, I didn't say that.
You did.
I meant it doesn't matter where you come from. It matters where you finish.
I resemble that remark.
Indeed. Who used to say that all the time?
Groucho Marx?
No, Daffy Duck.
He did?
Yeah, he did.
Oh,
that was one of his favorites. I can't do a Daffy Duck, so I won't do that one.
All right. Do you want to do a paper today, Dickson?
Of course.
All right.
But this paper requires
some background.
It does, because this is not your typical parasite paper. All right?
Although it's cool.
It's very, very cool.
And I should tell people you picked it.
I did. Because I'm a big reader of Science Magazine, and you claim that you're not, but I don't believe that. I think you really are.
I don't read any particular journal.
Right.
What I do is get a feed of all the virus articles that are published.
Right.
I am agnostic when it comes to the journal.
Well, I appreciate that.
I am not a big fan of luxury journals like Science, Cell, Nature, etc. I feel that they inappropriately bias the scientific literature because they only publish what they think is hot.
Yeah, no, that's I have to agree there because I tried many times to get some of my own work published.
Did you ever have a science paper?
I did. I have 2.
Really?
Yeah, 2.
Why don't we do one of them sometime?
No, please. No, at the time, actually, one was solicited by Science. They wanted to publish more parasite papers, and they went to the American Society for Tropical Medicine and Hygiene meeting, and they my graduate student delivered a paper at this meeting, and one of the editors for Science came up afterwards and says, "Would you consider publishing in our journal?" And we said, "We'll think about it." And no, we said, "Yes, sure, we'd be happy to," and we did, and we were very glad we did that.
You have 58 papers to your name.
Well, it's more than that, but that's all that's listed
PubMed?
here. Yeah, but PubMed doesn't have all the papers. Though.
Why not?
Because a lot of them have appeared in obscure journals. So they don't excerpt like Vitomyska Scientifica from Poland, for instance. And
so your first paper, according to PubMed, is 1967.
That's correct. It was on ferritin conjugated antibodies. Why do I know the title of that paper?
Yeah,
that was the first one. It was with Dr. Kojima.
Yeah,
Masahiro Kojima, and that we were doing an electron microscope study on the nematode Trichinella. Yeah, so
I'm looking for your science papers. Here we go. Effects of Trichinella spiralis on host responses to purified antigens.
Right, with David Silverstein.
He was your only student, right?
He was my only student. And I had two, actually. The other student was from Cuba, and she went back to Cuba to receive her degree, but she actually did her work here.
And the second title has a typo in it. Effects on Trichinella spiralis of host responses to purified antigens.
Oh, good. Right.
Should we do it instead of the one we picked?
No, please.
Because you don't have to read it. You should know it by heart.
A historical walk back in history type of thing. No, we don't want to do that.
It's actually tripping back in history.
Well, for me, it would be tripping. This article that I selected was based on two premises.
Pipette.
Number one,
with a pasture pipette.
Yeah,
he writes.
Number one.
Sorry.
It's not a typical parasite paper. It involves plants.
Plants can be parasites?
Plants can be parasitic. That's correct. And number two, it involves a lot of molecular biology. So I thought if I selected this paper, it would afford me the opportunity of listening to you wax eloquent with regards to the molecular aspects of this, because you are certainly much more of a molecular biologist than I ever was, and you're still a practicing molecular biologist that works on viruses. So this paper smacks of some of the material that we've already done on TWiP, for instance.
I pray at the altar of molecular biology.
Yes, you do. Yes, you do.
It is what makes everything work.
Well.
All right. So, what did you read the title of the paper?
Would you like me to?
No, I want you to, Dickson. You picked it. Read the title
here we go.
And the authors.
Let's see if I can read the title of the paper.
Why can't you?
Well, of course I can. So this is plant science. Plant science separates animal and plant science, apparently, because they've got a big red headline at the top of this paper that said plant science. So the name of this paper is Genomic Scaled Exchange of mRNA between a parasitic plant and its hosts. And it was intriguing, and I started to read the abstract, and the abstract was very compelling and told me things that I had no idea about before this.
Who are the authors of this study?
The authors of this paper are Gunjun Kim, Megan LeBlanc, Eric Wafula, Claude D'Amphilis, and James Westwood.
And where are they from?
That's a very good question. And they're from multiple places, but the senior author, who happens to be the last author on the paper, is from Virginia Tech, from the Plant Pathology, Physiology, and Weed Science Department.
Hokies.
They're not Hokies.
Hokies.
No, Hokies are from Oklahoma.
No, Hokies.
Oh, Hokies. I'm sorry.
Virginia Tech.
Yeah, they're the Hokies.
The Hokie.
They are the Hokies.
I know this because now when I go to a school, I learn what the
mascot. So what's a Hokie?
It's a bird of some kind.
Oh, I didn't know that. Well, Blacksburg, Virginia is a beautiful place, as you know, and Virginia Tech is really noted for its ecology. I knew a very famous ecologist there, John Cairns Jr., who for many years ruled the roost, so to speak, at Virginia Tech with regards to ecological studies.
There's another set of authors from where?
Right. The other set of authors are from the Department of Biology and the Huck Institute for the Life Sciences at Penn State University in University Park, Pennsylvania.
Nittany lions.
The Nittany. Now, what's a Nittany?
I don't know. What is a Nittany?
I have no idea.
It's a lion of some kind.
Well, what do they mean by that?
You want me to look it up?
No, you don't have to do that. The lion, in this case, is a mountain lion. So I think Nittany is the name of a mountain somewhere near
It's a mountain
Pennsylvania.
lion that once were fabled to roam near the school.
Exactly.
Mount Nittany is a local landmark.
Yeah, that's what I actually thought.
Nittany lions. Now,
right.
Dickson.
Yes.
This paper is about a plant parasite.
It is.
It's not something that burrows into the plant. Are there those kinds?
So let's back off a minute.
Take it away, Joe.
Well, there you go. So there are a lot of people out there that have been enamored with
Is
organic...
that like love with?
Enamored with organic cultivation of plants.
What does that mean, organic cultivation?
Well, that's right. What does that mean? But, permaculture, for
Not
instance.
using pesticides?
Not using pesticides or fertilizers or herbicides. Growing them naturally.
Isn't fertilizer organic?
Well, sure it is, but some parts of it are, and other parts are not. So that's
I'm being testy. I'm sorry.
a zero-sum game if you want to play that one. But the part that gets them the most is the fact that rich soils contain fungi. Mm-hmm. These are now useful fungi that create connections between the plants, and they're called mycelia. And so there's a whole book on mycelia called Mycelia Running. It's a sort of a takeoff on, the Bruce Dern movie of, transporting plants to outer space until the Earth comes back to normal. So the mycelia are the connections from the fungi.
Mycelium running.
Mycelium running.
How mushrooms can save.
That's right. How mushrooms.
The world.
So they're fungi, right? They're basidiomycetes. Basidiomycetes.
Oh, yes.
They're edible fungi. And so these have root systems. They're not really roots. What they are, because all fungi are saprophytic or parasitic.
What does saprophytic mean?
A saprophyte is an organism that eats dead things. So in the woods...
We're saprophytes.
Well, of course we are. We're not all of the time. If you buy fresh vegetables, those are still alive when you're eating
dead.
them.
Once you
Can't
pull
you
them out
hear
of the
them
ground,
screaming?
They're dead.
You eat a carrot and you go, oh, don't.
That's not. Are we truly saprophytes?
Well, we eat dead meat. You'd say that those plant cells are alive, but the animal cells are probably all dead.
For sure.
So we very few groups of humans eat living things.
So when you put a potato in the fridge and it grows...
No, it's alive.
That means it's still alive?
Of course.
So when I boil my potatoes,
You're killing those poor little things.
That's why I don't like to eat lobster. I hate killing them.
Well, I don't know what to tell you about that one. It's an individual ethic at this point. But the point is that people who grow their own food in their backyards and get involved in organic farming because they save all of their waste products from their plants, not the animal parts, but the plant parts, and they compost. Mm-hmm. And then they use that compost, which is filled with worms, to throw it on top of the soil before they plant their crops, and they get a much higher yield of tasty, wonderful vegetables. Now, I think everybody out there that listens to the TWiP and TWiV and TWIM knows that there's also this urban agriculture website that we've started, the podcast, I should say. And we've compared and contrasted outdoor agriculture with indoor agriculture. Indoor agriculture doesn't require any of these things that outdoor agriculture does. So the development of mycelium and a fungal root system that are connected together, which allows the plants to exchange nutrients through these connections that the fungi make between individual plants. That's true for a planted community like corn or sorghum or soybeans, but it's also true for natural populations like forests.
And this does not happen in hydroponics.
Not at all. In fact, you only want one thing growing, and that's the one thing that you've put there. So what it does ecologically is it connects things together and allows plants to share. That's the best way to put it. So the good fungal mycelia connect up, let's say, a hardwood forest. Okay, now you can learn this through ecology courses. And so that means that all of the trees of a forest, a given forest, share water and micronutrients through these fungal connections. It's quite amazing. It's like a gigantic colony. And in fact, you know that the world's largest organism is not the blue whale.
It's a fungus.
It's a fungus.
What is it, like 1,500 miles long?
It used to be, it's a circle. It's actually a circle, all right? It used to be, they used to think the one in Wisconsin was the biggest, but they found another one in Oregon, which was even bigger.
Right. They're underground, right?
The mycelia are underground, but the fungal bodies, the fruiting bodies, actually come up out of the ground. And those are the things that we cherish when we go wild mushroom hunting.
I don't do that.
If you know what you're doing, you can have a wonderful time. And if you don't...
Did you ever eat a mushroom that you picked?
Many times.
Well, I'm glad
Many
you're here
times.
to tell us about
No.
it.
Well, if you know what a chanterelle is, you can easily distinguish them from all the
Have
other
you ever
mushrooms.
eaten an Ammoniten phyloides?
Well, I wouldn't be here if I had, would I?
Ammonites phyloides?
Yeah.
How do we pronounce that?
Ammonitin is a dangerous substance. Doesn't that disrupt the RNA synthesis process?
Yeah.
That's
It's
what I
an
thought.
inhibitor of
Yeah, it's used-
DNA-dependent RNA.
it's-
Ammonita phyloides,
There
right?
you go. Yeah, the angel of death is
And
what it's
the
called.
toxic compound we use in the laboratory to inhibit RNA
This is ammonite.
synthesis.
That's right.
Ammonitin.
That's right.
Wow.
The answer is no, I've never eaten one, nor will I ever.
All right. Go ahead. Keep talking
No,
about
okay, so I
parasites.
wanted to make a connection between good fungi and bad fungi because you got some bad ones out there also.
Like what?
Wheat rust.
It's not good for your wheat crop, right?
If you look it up, you will see that it's not just decimating the wheat
No,
population.
decimate is not the right
It's
word.
1 in 10, right?
It's not good
Sometimes it
enough.
wipes it all out.
Yeah, exactly. Wheat rust is
And
a
it's-
fungal disease that affects wheat, barley,
Right.
and rye stems,
That's
leaves, and grains.
Right.
It is destructive because the pathogen overwinters.
This is correct.
20% yield loss.
Yeah.
Wow.
That's enormous
The
loss.
pathogen is Puccinia rust fungus.
That's right. And it's airborne. So you can't protect against it outdoors. You can't say, well, I hope the wind blows the other way tomorrow because if it doesn't, I'm going to lose all my wheat.
Spreads via spores.
It does.
Area.
It does.
Airborne,
Airborne.
like some viruses,
Got it right. But not all.
not Ebola,
No, certainly not Ebola. So,
not
So-
HIV,
no. But so rice blast.
not
and
hepatitis C
wheat-
virus. Sorry.
I'm sorry. I know we've had other issues that we've been discussing on another show, but so we mix and match here because really what we've got now is a crossover host-parasite relationship that we're going to talk about, and
Why
neither
do you
one of
call
us
it a
are
crossover?
well because neither one of us are plant experts.
I thought you were.
I'm not a plant expert, but I know enough about them to
be
take a
dangerous,
chance to
be
talk about this
dangerous,
paper.
right?
Some plants are really dangerous, and
You know
others,
enough to be dangerous.
Oh no, that may be true. So
Do
we
you
have
think
two
our
kinds of-
listeners want to hear about plant parasites?
Oh, sure they do, because I think everybody out there has to understand why, in some cases, there are shortages of the edible plants that you and I have come to expect in the marketplace.
So these plant parasites can impact
Of course they can.
the growth of the
course.
plant they're parasitizing?
There's a huge investment from the government through the United States Department of Agriculture in plant pathogen biology. They're hugely
Sure.
interested in this.
These
And-
land-grant colleges, right?
Oh, yeah, well, there's lots of them. I mean, almost every state has their own ag school, and every state certainly has their own IT school. And a lot of cooperation occurs between those two kinds of schools.
Does Massachusetts have an ag school?
Oh, yeah, the University of Massachusetts at Amherst.
Yeah,
It's
there's
got an
a school.
enormous agricultural
About
school.
Vermont,
Yep,
Maine,
I think so.
New Hampshire,
Absolutely.
Rhode Island.
I think all of them do.
I'm trying to trip you up. Connecticut,
If you go and look it up online, you
San
can find...
Marino.
Stop it. That's not a... You're okay. You win.
What did I win?
If you, I don't know. We'll have to determine that later. But if you go on the net and just type out United States agricultural universities or agricultural colleges, you'll find almost every state has them. And Rutgers, by the way, which is not too far away from you, where you and I live, has a world famous agriculture school. And the same is true for California. UC Davis and some other campuses there too. Cal Poly has a wonderful agriculture school. So, and those places are concerned with things like soil types and weather patterns and climate change issues. But a lot of people there are concerned about plant pathogens. A lot of them. And many of them, by the way, I don't mean to denigrate viruses at all, because a lot of plant pathogens are viruses. In fact, I know, Vincent,
Sure,
you
of
can
course.
tell us what the very first virus pathogen was that was identified ever.
Plant pathogen, you mean?
Yes.
Tobacco mosaic virus.
Bingo.
And aside from viruses, another
And
important...
it looks just like this.
Here, let me take your picture.
Take a picture of it.
I'll
'cause
put it in the show notes.
Here is tobacco mosaic virus represented.
Smile, Dickson.
Okay.
Very good. Dickson,
Yes.
aside from viruses, the other important pathogen
Yes.
of plants
Fungi.
is aphids.
Well, that's not a plant pathogen. It's a deliverer of pathogens.
Yeah, but don't they
It's a
remove
living-
the sap? And
Yeah, they do that,
that's
but
bad?
that's
Doesn't hurt the plants to remove the sap?
Well, some of them it does.
I mean,
of
if you have
course.
billions of aphids on a stem.
No, that's bad. That's bad. But even a few aphids or aphids, which have viruses in their hemocoels,
they deliver them.
Can, as a syringe, deliver that right in.
Okay.
So today we're going to discuss an experimental model for plant pathogen behavior. And the plant of choice in this case was actually two of them. One was a tomato plant, which has got high commercial value.
Sure.
And by the way, you know where tomatoes come from, Vincent? Do you know the country of origin for tomatoes?
No, I don't.
It's Peru.
Really?
It's where tomatoes and potatoes they both come from the same place. So and the pathogen that they're going to try out attacks not just tomato plants, but will also attack Arabidopsis. And
Arabidopsis, Dickson.
It's a small flowering plant which is chosen for its small genome size because a lot of plants have huge genome sizes. So Arabidopsis has been sequenced totally, and it's ease of handling in the laboratory.
It's also a weed.
But
it's a good model organism.
Yeah. It's a flowering plant.
Okay, Arabidopsis and tomato.
And the parasite in this case.
What's it called?
I'm going to read the name of this, and I'm going to completely blow it because of the plant pathogen people listening out there. They're going to get a kick out of the way I'm going to try to pronounce this. The common name for it is a dodder, Dodder.
Mm-hmm.
And the real name of this organism is called Cuscuta, pronounced Cuscuta or Cuscuta.
I would pronounce it Cuscuta, or if I were Italian, Cosciutta. But it's probably not the way they pronounce it.
You mean like prosciutto?
Exactly. Because we'll say Cuscuta.
And then Pentagona.
So
it's got five-sided organism. It's a five-sided organism.
So folk names for this include...
he's got it right up
Devil's Guts,
Nice.
Devil's Hair, Devil's Ringlet, Gold Thread, Hailweed, Hairweed, Hellbine, Lovevine, Pull Down, Strangleweed, Angel Hair, and Witch's Hair.
I like Strangleweed. That's a great name for this.
So how does this parasite parasitize a plant?
Okay.
Tell me.
Okay, here we go. There are unique structures that are produced by fungi of various kinds, and by some plants as well, called Hastoриum.
Hastoриum.
Hastorium.
Got it.
And what it is a cytoplasmic projection off of the root system or off of the mycelia of these parasitic fungi, which actually can penetrate the root systems of their host plants.
Mm-hmm.
But they don't actually insert themselves into the cytoplasm of the plant cell itself, because plants are arranged a little bit differently than animals, of course. They have cell walls in addition to cell membranes. So the membrane is on the inside, and then the cell wall, which is made out of cellulose, is the protector unit. That's why plants can assume such geometrically interesting shapes, because of these structural units that are made out of polysugars, of various kinds, all right? Like cellulose, for
Oligosaccharides, Dickson.
Oligosaccharides.
Polysugars.
Well, come on. We're trying to talk to a common audience here that doesn't know that.
Oligosaccharides.
Yeah, okay, oligosaccharides. Okay, fine.
Yeah, so, you're right, Dickson. Polysugars.
Yeah, they're polymerized chains of sugars which assume geometric shapes. Okay, so these Hastoria insert themselves between the cell walls.
And there's fluids.
And there is fluid. And there are two kinds of main ways that plants get materials from the soil, or from the interstices of the soil particles, I should say. Namely the water, and then the micronutrients, and then their food sources, which they, of course, store in their root systems in the winter and then reutilize in the spring and summer. Okay, and they all store it that way too. So there's two kinds of cells in higher plants. One is called the xylem cells, and the xylem, the way I learned this in high school, and everybody else did, I'm sure too, is that food flows through the phloem, which is the other set of cells, and water zips up the xylem, because xylem starts with an X. So there's no way to forget that once you've got that little saying under your belt. So it's complicated, though, because there was a PNAS article, which I also recommended to you, to show you the electron microscopy of a parasitic fungus making a hostorial connection with its plant cell. And it's not obvious, even by looking at that wonderful cross-section of the fungus as it inserts itself below the plant cells and then creates this little bladder below it as to what the actual connections are. And in fact, this article also says that we're not actually sure as to how this exchange occurs. So the first thing to know is that they make a connection, and they make it through their hostorium. The second thing is, what are they connecting to? And what are they deriving from the host? And what is the host deriving from them? Now, if you look in
the host derives something from
yeah, because if you look in the woods, right, and this mycelium that develops between the good fungi, they are some people have referred to them as the internet of the forest.
Yeah, right.
And others have referred to them as the nervous system of the forest. But in essence, what it does is it allows a trafficking of substances between individual plants. And in this case, they're trees. The ones that I'm most familiar with are trees. A parasite just wants to derive something, right? And it doesn't really want to give anything back because apparently they need more than they can give. But plants are kind of borderline in terms of that philosophy. And in fact, there may be some modifying substances that plant parasites need to give to their hosts in order to get their host cells and tissues to give up more back to the parasite. So this article addresses the issue of exchange rates of molecules between a host plant, in this case either a Arabidopsis or tomato, and the fungus, this...
Not a fungus.
Well, the Hostoria.
The Cushuta.
Yes, that's right. It's a dodder.
It's a plant.
It's a plant. Yes, it's a dodder.
Fungus. A dodder is a plant.
You know, okay, fine.
It's a parasitic plant.
But no, I understand that part. But the connection between this parasitic plant and its host cell is the same.
Resembles as that between a fungus.
That's right. That's right. So this is a bigger organism that they can now use.
So many people might have seen these parasitic plants. They wrap around the stem of the plant.
That is correct.
And the little hairs insert into the stem.
Hostorium.
Hostorium.
There you go.
And then they remove nutrients.
That's right.
Now,
Or something. And that's the point of this article. What is it?
So the thing is that's cool here. Let me just give you a little background
Please
here.
do.
So a dodder grows on its own. It has a root system.
Okay.
And then it finds a plant. And do you know how it finds the plant? There's some evidence that it senses organics put off by plants. And
it follows gradients.
And here's a plant. So then it attaches to the plant. It wraps around it. It makes a beautiful coil.
Right.
Right?
Yep.
And then it inserts these hairs. What did you call them? Hostorium.
Hostorium.
And it begins to derive something from the host, and its root system goes away. It says the original root of the dodder then dies, and a dodder then thrives on this plant. Sometimes it will make the plant ill because it's taking, and it makes it more susceptible to diseases like virus diseases. It can also attach to multiple plants.
Indeed.
So there could be an interplant. We have an internet. This could be an interplant.
Do you know of any examples in nature that you could liken to this? No pun intended.
No pun intended.
None.
Lichens?
Like a strangler fig. Do you know what a strangler fig is? Wow. The Brazilian jungle, well, most jungles are alive with these plants. They're actual higher plants.
Oh, yeah?
They're figs.
Here's a picture of one wrapping around.
Neat.
And then here's a whole tree full of them.
That's fascinating.
This is in Punjab. Do you know where that is?
Yes, it's in India.
Yeah. This is this Cushuta on an acacia.
An acacia?
You know what an acacia is?
It's a tropical tree.
This is pretty cool. Volatile substances.
So they're actually attracted to gradients of host-derived materials.
Now, Dickson, this is a true parasite because it harms the host.
That's right. No,
Isn't that part of our original definition way back in episode number 1?
Yeah.
It harms
it is a parasite.
The host.
That's right. That's right.
Right. Do the fungi harm the plant when they attach to them?
Not the friendly ones. The friendly ones are the ones that make the connections for the other plants.
Those are mutualistic relationships.
They are mutualistic.
This
is
correct. A parasitic.
This is exactly right.
Okay. Cushuta.
Right.
Cushuta.
Right.
So in this paper, Dickson.
Yes.
Previously, there was some evidence that something is going back and forth between these two plants and the parasite.
Right.
And they use modern DNA sequencing
They do.
to study this.
Yes.
Deep sequencing.
Very.
Next generation sequencing.
And billions of transcripts.
Billions and billions.
It's incredible.
Like Carl Sagan level
Exactly
transcripts.
Right. I was amazed when I read the details of this article to find out how sophisticated and how advanced plant sciences have come. They were being pulled and shoved and, sort of marginalized by most of the people involved in molecular biology. But
That's right.
plants actually are central to the life of all animals.
So in the lab, they grow Arabidopsis
Right.
and tomato
plants. They're easy to
and
grow.
then they let the cuscuta wrap around it.
That's right.
Then they cut 3 pieces. They cut a piece
Right.
of just
Right.
the plant.
Right.
They cut a piece of
of
plant
just-
plus cuscuta,
And just the cuscuta.
and then
That's
cuscuta.
right.
They nip, snip, snip, snip.
Yep. And then they smash it all up and look
Smash
for the
it all up,
messenger RNAs.
And they
They
purify.
do CNAs,
They
cDNAs.
purify RNAs. They
That's
convert
right.
it to cDNA,
That's
and then
exactly
they deep sequence
yeah,
those.
yeah. Deep sequencing.
Deep means
That's
lots
amazing.
and lots and lots,
Amazing.
millions and millions
Amazing.
and millions of
Amazing.
reads.
Amazing.
And they ask, so they know the genome of the transcriptome, I
They
should
do.
say.
And
The
they just did
transcriptome
tomato, by the
is
way.
the total
Right.
RNA of the plant. Yes. So in the process of doing this, they had to do the tomato
Yes.
transcriptome. They
That's
did the Arabidopsis transcriptome was
Yep.
already known.
Yep.
So they can say in the plant, in the mixture, and in the parasite alone,
Yeah,
what kind of
yeah.
RNAs do we find, right?
Exactly.
Now, where you have the two wrapped around each other, or I should say the cuscuta wrapped around the
Right.
plant, they find
Right.
50-50 more or less, which is
Yeah.
what you'd expect, right? Because
Yeah.
you're sampling both tissues.
Exactly.
That's kind of a control, right?
It is.
Now, if you look
But-
at just plant,
But
if you they have these nice pie charts that show this. If you look
see
in
that.
Arabidopsis, you get about 0.6% up away from where the cuscuta is hooked. 0.6% of the reads are from the cuscuta.
that's amazing.
And if you ask, if you look in just the cuscuta, you get 1% of tomato Arabidopsis.
Understood.
Less of tomato. The
Right.
tomato cuscuta
Right.
don't seem to exchange as well
Not as well.
as the Arabidopsis.
That's right.
But they do. It's amazing.
That's incredible, isn't
Now,
it?
1% doesn't seem like a lot, but it turns out there are thousands of mRNAs.
So what is
involved
this
there.
parasitizing?
Well, it's the thing is, it seems to be going both ways.
And what are the messages? What do they translate into in terms of proteins?
Oh, gee, that's a good question.
I mean, that would be the next thing to know, wouldn't it?
Well, one of the things that they find is that the most highly abundant mRNAs tend to be exchanged.
Right.
But not everyone is highly abundant.
Right.
Because if you say it's the most highly abundant, then that's just, wow, those are the ones
But they had
most
some
likely to
anomalies
get
there.
in.
They
But
had some anomalies.
there is some lower abundance one that gets in, but so that doesn't really
Right.
tell you.
Right.
Is it associated with gene function? Well,
Ah.
they do some kind of cluster. They do gene function analysis, and they say these are mRNAs that are exchanged. It's not all of the mRNAs, so is
No.
it some particular function?
Right.
Well, some of them are involved in mobility, intercellular mobility.
Right.
Okay. So they say, so here, for example, the mobile genes, 36% of them are involved in metabolic processes. 31% are involved in responses to stimuli. 20% hydrolase activity. Do you know what hydrolase is?
Sure.
What is that?
Well, when you enzymatically split something, it produces water as a result.
Now, non-mobile components, some of them are involved in biological regulation. That wasn't represented in the mobile one, so that's
Yeah.
kind of interesting.
Yeah,
Catalytic
yeah.
activity. And metabolic processes, which also were mobile, many of those are non-mobile. So basically, Dickson, this doesn't tell you a lot because it's just everything.
Oh, that's right.
A lot of things
So
are
how would
moved.
you investigate this as a biologist? Just stop right there and say, I don't want to read anymore. I would just like to think about this. And what would I do next as a biologist to try to sort out what these things are doing?
The problem is, Dickson, that they're many transcripts that are exchanged.
I know. So you have to start somewhere.
8,655 Cuscuta genes were classified as mobile into Arabidopsis.
That's incredible, isn't it?
And similar with lower, but
Yeah.
same idea with tomatoes. So it's a lot.
I was also stunned to see how many genes there are involved. I mean, Arabidopsis has like 35,000 some odd genes.
Here's the hypothesis, that some of these genes from the parasite going into the plant have a negative effect, right?
Right.
Or maybe they stimulate something that they want. Who knows?
Or maybe they're just sharing resources.
Why would a parasite share resources? To
Well,
allow
maybe
the
we've
plant to grow?
misnamed it. What happens to the productivity of the plant that it parasitizes? You know, with tomatoes, you could actually tell.
Do they go down?
I don't know.
Do they get smaller?
I think they do.
So personally, I don't understand why a parasite would put mRNAs into a plant. All right? Unless it wants to increase something that it wants.
Right.
Or it just want to kill the plant, right? Because
No.
that would be counterproductive.
No.
Right? So maybe it wants to set up some kind of transport system to take things away, right?
It's hard to know. I mean, animal cells that grow next to each other, they don't exchange messengers. That doesn't occur. Animal cells do not behave this way.
You know,
Tissues don't behave this way.
They can exchange small regulatory RNAs.
Really?
And there are things called exosomes that vesicles that bud off the surfaces of animal cells, and they have RNAs in them, and they can exchange them this way. So I don't think it's, Dickson, we never say never in biology.
This is true, but this is a new thing for me, at least. I mean, I was totally unaware
One of the first things I would do is see if any of these mRNAs that are exchanged are translated into protein.
Right.
Because if they're not,
Right.
well, it doesn't mean they're not important, but it brings you down another avenue of inquiry.
Yeah,
if they're
that's
not
right.
translated, then they may have regulatory functions. They may
Exactly.
be regulating genes.
Exactly.
But 5,000 or 8,000 is too much to take one by one and figure out what they're
And
doing.
do knockouts. You couldn't do
Can't
that.
do knockouts.
No,
That's
you
very
can't.
difficult.
That's
and,
right.
I mean, you could try injecting the RNAs one by one into the plant to see what the result is, but very
I wouldn't want to be that
difficult.
graduate student.
Very difficult to do.
Absolutely. But there was an opening statement made in the article that ordinary plants without parasites, those cells exchange messenger RNAs between them. So there's a lot of interplant exchange of messages. Is this to homogenize the growth rate or the growth systems of plants? I mean, to get everybody on board, so to speak. Okay, today we're going to make these proteins. Everybody wake up. And then they send out a whole bunch of message, and all the plant cells take them up, and they all start making the same proteins. Is that a one way of looking at this system, as opposed to, let's say, a multicellular organism like a human that has so many different cells that it's hard to keep track of all of them?
It's a good question. We don't
And
know.
another question
No
I have,
idea.
another question
That's
I
why
have
it's a science paper.
is, what is the effect of the microbiomes of these two plants on each other and on their expressions? And do they have a virome also?
Of course they do.
They have to. So you've got these complexities upon complexities going on. I know Arabidopsis can be grown germ-free, an agar with nutrients in the agar, and that's how they handle this in the laboratory. But I don't know whether they're actually bacteria-free or not. I don't know. Don't have any data on that. So it's an intriguing...
So the real question is, what are these exchanged
yeah,
RNAs
that's
doing?
right. What are they doing?
So the parasite may be receiving some from the host, maybe they
Right.
function in it, and conversely, it may be putting things into the host to regulate it in some way.
Exactly.
But how that happens, whether it's through protein or RNA, regulatory RNAs, we don't know.
This data is overwhelming in terms of its
They
scope.
clearly demonstrate exchange. Now the next
Sure,
question is
sure.
whether it is of any significance.
That's right.
And that's we don't know.
That is exactly right.
The other possibility they bring up
Yes.
is that this could lead to gene exchange,
An even
horizontal gene transfer, HGT.
And that sounds like
Okay.
a virus job.
It can be a virus job. It can be done by mobile genetic elements like plasmids.
Yeah, plants are famous for that.
But this is RNA, Dickson. Typically, we think about DNA moving
I know.
from cell to cell.
I know.
So if this, in fact, were a way of gene exchange, what would have to happen to this RNA before it could get into the genome?
What if an antisense RNA molecule got into a plant cell and then reverse transcriptase made the DNA molecule of it and it inserted into the genome?
Plants do have reverse transcriptase.
Ba-boom.
So that's what could happen. These mRNAs could be copied into DNA, and then that could integrate into the plant cell genome.
Right.
So I would like to know whether there are any genes that are inherited, say, from Cuscuta in any of the variety of plants out there that normally parasitizes.
That's right.
Right?
That's right. Now, another interesting facet about plants in general is that they don't exactly have an immune system that you could liken to an animal immune system. So, for instance...
But they do have RNA interference to defend themselves.
They do, but Luther Burbank made his living hybridizing one kind of plant on another kind of plant by simply making a little cut in the branch of one and inserting the branch of another one in it and then wrapping it all up, and the next thing you know, they're growing together.
Why did he do that?
So that you could have apples and pears growing on the same tree. Now that's really unusual because the parent organism is the apple tree, let's say. Mm-hmm. But you'd say, I would like a half and half tree. I would like a tree to grow pears and apples for me.
Right.
And the next thing you know, you've got an apple tree growing pears also. Or you could hybridize...
Apple Trees Productions.
Let's...
LLC.
If you look at what a nectarine is, or if you look at some of the hybrid plants like broccoli, for instance, you end up concluding that plants are really accepting of each other. I mean, they will...
Yeah,
okay.
you're applying a human... feeling to this.
But only
you're anthropomorphizing.
No. I'm just comparing and contracting.
Dixon, the last thing I want to bring up.
Sure.
So this movement of RNA could result in gene transfer from plant to Cuscuta or back.
Yes.
But since Cuscuta can bridge plants, could also take an RNA from one to another.
Exactly.
So this could contribute to diversity in the plant kingdom by exchanging genes and letting plants adapt to different environments, right?
So the plants also have another interesting arrangement of their genomes, and that is most of them are ploidy, compared to this 2N for most
not just diploid, they're
not
multiploid.
Just diploid, they're tetraploid.
Tetraploid.
That's right.
Are they aneuploid?
I don't think so.
Do you know what that means?
You don't have a nucleus at that point.
Aneuploid means that you have... Every cell has a different number of chromosomes.
Oh, I see what you mean.
Right?
Okay, so... I was told once that the organism on Earth that has the largest, most complex genome is the orchid. I don't know if that's still true because I know a lot of genomics have been done since then.
Yeah, that rings a bell.
But maybe we could find that out for the reader, or the listeners, I should say, to see whether or not orchids still hold the world's record for
the largest genome.
The largest genome, right?
Well, let's see.
World's largest genome.
All right.
Who won that contest?
The smallest. What's the smallest genome? Do you know?
It must be a virus of some sort.
Porcine circovirus. Well, actually, it's the viroids are even smaller. They're 300 nucleotides.
See.
All right, the biggest genome.
Yes.
Vertebrate.
Really?
Fish.
Oh, okay.
It's a fish. The marbled lungfish.
Really?
130 gigabases.
Wow.
The mammal genome, mouse genome, is 2.7 gigabases, 2.7 billion genes. Okay.
Wow.
So 130 billion genes is this marbled lungfish. Humans are 3.2 gigabases or 3.2 billion base pairs. Okay.
Yes.
So it's this fish.
But what about plants?
Are there any plants on this list? Let's see. Yeah, Arabidopsis is only 157 million base pairs. Right. The orchid, let me see if I can find that. I know that was a big one.
Yep.
It's not on this list. Let's look it up. Size of orchid genome. Genome size. Overview of the orchid genome. 1,600 million, so that's 1.6 billion. So it's pretty big for a plant. Well, I shouldn't be judgmental, Plants can be smart, too.
Well, they can certainly be pretty because look at the orchids, right?
Haemophilus influenzae. 1.8 million. That's the first genome of a living organism sequenced in July of 1995.
Bingo.
The largest known viral genome, Pandora virus salinus, 2,470,000 base pairs.
And as you continue to climb the phylogenetic tree of life, get into Neurospora, I presume, and things of this sort, and then or yeast.
Saccharomyces is 12 million. base
Right.
pairs.
Right.
Aspergillus is 30 million. Caenorhabditis, 100 million.
Look at that.
Silk
Drosophila.
moths are 432 million.
But it doesn't tell you much about the way it functions, though, does it?
No, it doesn't at all. You know what? In the human genome, 3.2 billion base pairs.
How many?
Only
are-
1.5% code for proteins.
Look at that.
Dickson, 1.5%.
I hear you.
Let me say it one more time. No.
What was that number
The
again?
rest are introns
Yeah.
and
And repeats.
retrotransposons
Yeah.
and other kinds of transposons. It's really remarkable.
It's befuddling to come up with
All
a
right.
function for all of that.
Do you want to read some email?
Sure. This was a great article, by the way. But I think it left the audience wanting more information on this subject. Which the only way to do that is to have someone on that actually knows about this next time.
Well, that's okay. We are whetting the appetites of
We
the
are.
listeners.
We are doing
Giving
that.
them, and if we could do this more often, Dickson,
Well,
we could wet them more often.
That's right. We need to water this more.
You're going to read the first one from Andy?
I would be glad to. So Andy writes.
Just remember, don't read his last name, okay?
No, I didn't.
Okay.
Dear professors, I have been following TWiP for several years. I am a software engineer, and parasitology is only an avocation. As an undergraduate in the early 80s, I discovered my love of history of science and ecology, although I continued the path of a software developer. As an adult, I read Carl Zimmer's Parasite Rex and became totally fascinated with parasitology. Later, I read The Coming Plague by Laurie Garrett. What I love about both Mr. Zimmer's and Ms. Garrett's writing is how they tie the science to the scientist. I look at The Coming Plague as a masterpiece in history of science with really touching stories of the scientists and their impacts on the world. I'm writing to ask you to make sure that when you talk about the science of parasitology, you keep including the stories of who figured out what and why it was important. The two of you bring a lot of experience in parasitology, microbiology, and virology, and have lived the history of it. Keep the stories coming. If you haven't watched this, I recommend that you do. It's a lecture by Professor Walter Munk on his life's work in the field of oceanography. Fascinating. Thank you. P.S. I also recommend the book Krakatoa, The Day the World Exploded, as a great book with history, anthropology, and science all tied together with history of science. Thank you, Andy, for writing.
You know, the best science writers write about the people who do the science.
Yeah, no, I agree with you there. I do agree with you
And that's
there.
why Zimmer and Garrett's books are good.
No, I got it.
We try and bring people in, right? That's the way to
That's true.
make the science
This
interesting.
is
Of course,
true.
in the case of today's paper, we don't know the people, so we can't say
that's
anything.
Right. That's right.
I used to love Krakatoa when I was a
Ah,
kid. Did
me
you know
too.
that the island has become recolonized by plants brought along on the wind?
Sure,
Are you
of
surprised by that?
No, and neither was Charles Darwin. That's how he actually figured out that it wasn't one big giant continent that sunk and then you had mountain ranges and stuff like islands, but rather it sprang up from the ocean as a result of volcanic activity. So that's where his Galapagos hypothesis really shone through.
Next one is from Mike, who writes, Bobby Pritt was a superb guest. As Vincent commented, now we get to hear the pronunciations of the parasites, or something along those lines, which I also found helpful.
Right.
I had almost given up on this podcast because
no.
it seems to lack focus and rambles a bit. Hope you are able to get more people like Bobby on your show. Mike in Oregon.
Thank you, Mike, for pointing
Dickson,
that out.
are we rambling?
Sometimes.
All right. Let's just stop right now.
Not all the time. No. That doesn't mean we should stop doing what we're doing, because a lot of other people enjoy those rambles. But I understand what Mike is saying, and I think that it depends on who we have on as to what happens on the show.
Oh, by the way, I met a woman in Georgia,
Yeah.
University of Georgia, who works on malaria, and in particular, how the placenta defends against it.
Oh, nice.
And she would love to come on the show.
Let's invite her.
So we'll have her, and she can talk
Let's do
about
that.
it.
Let's do that. And some of our other earlier raconteurs, so to speak, particularly Bob Gauds and Tony James.
There are more people we can get. Sure.
Come on, we're working on it the whole time.
There are a couple of people right here in the city we'll get. We'll get
bet.
more people.
You
It's
bet.
just that, it's easy for you and I to sit down and chat.
And we like doing that, so that's probably why we
All right,
do it.
go ahead for the next one, Dickson.
I will. So, Kiri writes, Hi, Dickson and Vincent. Recently, I got a wonderful pug puppy from a local puppy mill store, which I know no one should ever do, but I had just lost another dog that was with me for 12 years, and I walked by, but not a puppy trap, not a rational state. No, I'll read that again. Who was with me for 12 years, and I walked by, not a puppy trap, not in a rational state. I think what she means is she walked by a puppy store and was tempted to go inside.
Yes.
The puppy, it turns out, has demotic mange. I had read about an oil from India called Neem that by some mechanism blocks the reproduction of bacteria, parasites, fungi, and everything. I put it on my dog, and weird white things started coming off within a half hour. I dusted her off, and they came back again 4 times. What is that? I also washed her twice a week with borax. I think she meant borax. And vinegar. What do you know about neem oil and its effects on parasitic life cycles? And what can you tell me about demotic mange and their behavior? Anything would be helpful. The dog I bought is super cute, but has many health issues, so I'm trying the natural path to reduce complexity. Also, a side note, Vincent, you are a wonderful and fascinating, but why so hard on your good friend Dickson? Yeah, why is that, Vincent? Believe me, Kiri, I don't take it personal. He seems like such a sweet person. Sometimes I want to yell at the podcast, leave him alone. No, this is fine. We're actually very, very good friends. By the way, I listen to all the TWiP podcasts as I go to sleep every night. Best science podcast out there. Thank you and all the wonderful people on the show for giving the lay people a little real science. As a side note, I am a software developer that has zero to do with microbiology outside of the fact that it affects us all. Thank you for the peek into the very small universe. It's really fascinating. I am so grateful. Thank you for everything you do, Kiri. I don't know much about demotic mange. I honestly don't.
I'll tell you what neem oil is.
Tell us what neem oil is then.
Neem oil is produced from the seeds of a tree, or of the neem, which is Asdiaracta indica, an evergreen tree endemic to the Indian subcontinent.
Uh-huh.
Okay, neem oil has a variety of fatty acids in it, basically. And some people may recognize them as the omega oils, right? Linoleic acid, oleic, et cetera, et cetera. Anyway, it's used for cooking.
Right.
And Ayurvedic uses include...
Which is traditional medicine?
Treatment of a variety of conditions, including fever, leprosy, malaria, tuberculosis. It is supposedly a contraceptive, a parasiticide, and an insecticide. It's used to treat a lot of things. So when something has so many indications, you have to be suspicious that it's actually doing something. So in terms of its antibacterial properties, but if it worked for your dog...
Exactly.
That's all that matters.
But the white things that came off, you'd have to be more descriptive in what they were, actually. Were they maggot-like things? Were they alive? Was it just white flakes of material, maybe some skin that was sloughing? It would be hard to know what was happening.
You don't want her to send them to you, Dickson.
No, I don't. But if she were to photograph one of them and send an email to me, I would be happy to help her identify it.
Wow, that's a good offer, Dickson.
I think it is. I've seen a lot, and I know people who have seen more, so we could get an expert opinion on that.
All right. Next email is from Rebecca. Hello, doctors.
Right.
I have just started listening to TWiP and am on episode 20 currently. I had a couple of things I wanted to share, and I apologize if these are things you have already covered in more recent episodes. First, in the episodes regarding Giardia and Entamoeba histolytica, it was stated that these are not reportable. Being a medical laboratory scientist, once called MedTech, I do report these to the local health department when we recover them. I live in Michigan, so maybe the regulations are different where you are.
Right.
I have attached a copy of the MDCH reportable diseases for you. Also, so I think it's not nationally reportable like at the CDC level. Well,
we meant to the CDC. Yeah. The CDC doesn't require knowing...
but at state levels...
Some states do, because they have control programs for them and they want to know if they're working. That's right.
I think some others have sent in state reportable disease lists to us.
That's right.
Also, when I was an undergrad, I read an article about a vaccine that targeted the gut of ticks.
Right.
So that when the tick took a blood meal from an animal, the antibodies would be ingested as well and kill the tick.
Exactly.
This reduces the amount of ticks in an area and the overall tick-borne diseases. I did a quick search. And sure enough, this research is still going on. What do you think about the possibilities of a vaccine similarly against, say, mosquitoes? Ticks take a larger blood meal, so it might not work, but it is still an interesting idea. Here's a link to the current work. So she sent a link to a paper in a journal called Parasites and Vectors.
Cool.
Have you ever published in that?
Nope, but I know one of the editors-in-chief.
It's called Antidote, an anti-tick vaccine to prevent tick-borne diseases in Europe.
Nice.
And this is a tick that transmits Ixodes ricinus. It transmits
Lyme disease,
bacterial, protozoal, and viral pathogens.
That's right.
This antidote is a European Commission-funded consortium of 7 institutes.
Nice.
Which aims to identify and characterize tick proteins involved in feeding and pathogen transmission.
Nice.
Dickson, do you remember our TWiP, and you will get to this, it's a later TWiP, Rebecca, where we talked about making antibodies against salivary proteins of mosquitoes.
Correct.
Right?
Correct.
So that when it bites the host, those proteins are neutralized, right?
That's right.
So, Rebecca, you'll find it. You'll get to it.
It's not a new idea either, because when I was at Rockefeller working in Dr. William Traeger's group, he had already thought about that a long time ago and had tried it several times, not successful, but at least the idea was there, and it certainly has some validity.
I absolutely love the show. I also teach microbiology to medical laboratory technicians at a community college, and I am going to recommend this podcast for them to prepare for their board of certification exams. So, Dickson, you better get it right.
You know that for sure.
I'm sorry. I'm giving you a hard time. Thank you again for the fantastic show. Now, I forgot to respond to the accusation of the previous email that I give you a hard time.
You just try that, if you will.
You know why I give you a hard time?
I don't know why. Do you give me a hard time?
Because I want you to get with it. I want you to do your work and be correct and precise. All right?
I try my best.
That's why I give you a hard time.
I try my best, Vincent.
You got to try harder.
I'm always on point here.
I make mistakes. It's fine.
I do too.
But
listen, I admit them.
Just get with it, Dickson. Dickson,
I'll try harder.
Can you take the next one?
Of course I can.
Erica?
Yeah. Erica writes, "Hi, I'm a zoology graduate currently working in a parasitology lab and applying for molecular parasitology PhDs in Ireland. Recently found your podcast and really enjoy listening to it in the lab. Wanted to say thanks. I was surprised at the lack of parasitology podcasts out there, and I'm glad you have covered it. Would enjoy it if you delved into some parasites of agricultural importance, such as PPNs."
You're going to have to figure it out, Dickson.
Well, I have a feeling that this is going to relate to some plant pathogens. That's what I think it is.
Plant parasitic nematodes, Dickson.
There you go. There you go.
We haven't covered any, have we?
No, we have not.
Can we?
Oh, sure. No problem there, because they're very important for certain agricultural crops.
You're going to need to do a little work here, Dickson. You're going to need to look in the literature and find some papers on PPNs.
Yes, sir.
Okay?
I will do this.
I know it's not vertical farming.
No, it's not.
Can you do this?
It's horizontal farming.
Alex writes, "Hi, guys. Love your TWiP. Dickson, you mentioned in TWiP 76 that H. polygyrus is the best model we have for human hookworm.
Right.
You might be interested in this paper attached that we recently published, which highlights how similar Nipostrongylus and Nicadar really are in terms of their biology and secretomes. I now think of Nipo as a better model for human hookworm infection than H. poly.
Okay.
Keep up the great work on the show. Thoroughly entertaining and informative.
Great.
Cheers, Alex." Alex is a professor
Indeed.
at the Australian Institute of Tropical Health and Medicine in Cairns, Dickson.
Cairns, Australia. That's right.
So he sent a paper, which I'm going to now tell you about.
Please
Dickson,
do.
The title of which is Secreted Proteomes of Different Developmental Stages of the Gastrointestinal Nematode Nipostrongylus brasiliensis.
Right.
This is from Alex's laboratory.
Right.
All right. And from this, he thinks that the secretome, that is what is secreted. Right?
Yeah. No, I'm aware of that one.
That it looks
Trichinella has one too.
more similar to
hookworm.
Yeah.
Okay.
These findings validate the use of N. brasiliensis as a suitable parasite for the study of human hookworm infections in a tractable animal model.
So some people will say out there, why don't you just use hookworm? And the answer is, of course, because it doesn't infect animals.
Yeah.
But But there is a hookworm of dogs, okay, called Ancylostoma caninum, which does infect dogs, and which is exactly a lookalike for the human hookworm species. But dogs are expensive to use, and I think they approach the limit of tolerance for the animal rights people that just don't want animals used at all. And so if you have to use an animal, use a mouse because I don't like them, but don't use a dog for God's sakes. Those are my pets. So we get a lot of that attitude also in biology, and it's hard to make it's hard to keep a balance in that thing. Okay,
last one, Dix. It's the last one.
Okay, the last one.
And then we close down the show.
Robin writes, Dave from Fresno, that's where I live, in your emails suggested that DVT may embolize to the brain. If it does, it would indicate an atrial septal defect of a ventricular septal.
Or a.
Should I try again?
No, just say or a ventricular septal defect.
You asked me if I could read this, and apparently the answer is no.
Can't. And I'm going to be accused of giving you a hard time again.
No, you're not. I won't do that. That's for sure. Or a ventricular septal defect. The emboli are too big to traverse the pulmonary capillaries. Also, aspirin and other antiplatelet agents are primarily effective against platelet thrombi, which are aggregations of platelets, like snowdrifts at sites of rapid flow, viz. Arteries. Such thrombi are friable, adherent, and less likely to detach en masse. DVT is more akin to a blood clot in a test tube. Multiple ring forms of the malarial parasite in an erythrocyte is indicative of Plasmodium falciparum infection. Well, I knew that. And then you should say, but did you know that...
Probably didn't properly express it, Dixon.
I'm thinking
this is
about...
what I'm telling you.
No, I'm thinking about these ads for Geico. Someone will say, did you know that... Yeah, but did you know that... And some of them were very funny.
Dave from Fresno previously wrote about... So I was talking about my long flight to Australia, and you were talking about DVT, deep vein thrombosis.
Oh, you were talking about...
Yeah, but you said it would embolize to the brain, and that's not right.
Did I say that?
Yeah, you did.
No, I didn't mean to say
I didn't.
that.
No, Dave said it, not you. Sorry.
I didn't mean to say
wrong.
that.
Dave from Fresno said it could embolize.
I don't have an
to the
opinion
brain.
on that subject. Thank you. No, it goes to the lung fields. I would say that's where you're most dangerous for pulmonary embolism.
And he said aspirins aren't going to help with those.
No.
And Dave had
No, that's
suggested.
right.
So but since you guys both live in Fresno,
You guys work it out.
work it out. Thank
Exactly.
you. Anyway, we appreciate your letters, your questions, and comments. You know where to send them, Dixon?
To twiv.tv.
TWiP?
TWiP.tv, sorry.
No, it's not even
WS.
close. This is why I give you a hard time.
All right, no. You're sitting there with all the information in front of you. I don't have to read that
You've
part.
been doing this TWiP.
I haven't been committing this to memory.
And you still, every TWiP I say, send your questions and comments to twip@twiv.tv. Now, why can't you remember that? See, this is why I give you a hard time.
Did I not say that? No, I didn't say the whole thing.
You said
That's true.
some Fakakta thing.
No, I said twiv@tv.
twip@twiv.tv.
Let's repeat that. Where would you send that comments?
twip@twiv.tv.
Thank you.
Dixon, where would you send questions
I
and
would
comments?
send it to where you just what he said.
Never mind. Oh, dear. This podcast can be found on iTunes and at microbeworld.org/TWiP.
Right.
And you can also find it on any of the podcatchers that you like. Since we started doing these podcasts, it used to be that you would find them on iTunes.
You usually do say that.
Where they still are. But now you can use a podcatcher, which is a little app that you can use to subscribe to a podcast, and it will automatically download them. Used to use iTunes, but now most of our downloads...
I think I'll enter the 21st century and get one of those.
Sure. I believe that when I see it.
Oh, you can help me do it after we hang up. We'll do
pigs
it.
will fly.
Oh, listen, if Ebola can become... No, that's not
Dixon,
go there.
do you listen to podcasts?
Sometimes.
How do you listen to them?
There's a dead silence in the room.
There's some buzzing noise, which has been...
There is.
What do you think? Is that someone doing...
dental work.
Sonication. Yeah, someone's sonicating.
Right. It sounds like a dentist.
Or maybe that's the construction site.
Always reminds me
anyway,
of that movie.
microbeworld.org/TWiP.
Yes.
iTunes,
yes.
twip@twiv.tv.
Right.
Thank you, Dixon. Dixon, I really appreciate your participation.
I Well, so do I. I mean, I appreciate the fact that we can still do these things together. We're up to number 77. That's pretty good.
Dickson can be found at verticalfarm.com.
Yep.
And I know I give you a hard time.
Racaniello.org.
Because I expect more from you.
My father used to tell me that all the time.
Okay.
And I did, but unfortunately he died before he saw me giving more than I could before.
I'm sorry.
Yeah, me too.
I'm Vincent Racaniello, and you can find me at virology.ws. Don't cry, Dickson.
I'm not. This is tears of joy here.
The music that you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is
parasitic.